Dr. Kert Viele, who is Director and Senior Statistical Scientist with Very Consultants. So Dr. Viele has a PhD from Carnegie Mellon University. He joined Very Consultants in 2010. He's been a PI on several NIHS funded grants, and he's going to talk to you today about adaptive trials and trial simulation. And I think there's a very great discussion of this that I think really breaks it down to an audience and makes it clear. So it's something that is definitely popping up more in clinical trials, and I think this is something that, whether it's relevant to your proposals now, is something that you will definitely run into in the future if you continue working in clinical. So, Kurt.
All right. Thank you for the introduction. Thank you all for having me. So I want to talk really about a couple separate topics. I'm going to talk about adaptive trials for a little while. Jason talked a bit about dose finding adaptive trial on Monday. I'm going to give an example of an Ebola trial that we designed. And then I'm going to switch over to trial simulation. Adaptive trials, that part of the discussion is really about what we're doing and why. And then the simulation is more about the how. How do we know that it works? How do we make sure that we're picking the right endpoint? Disclosures, we have multiple clients. I'm not talking anything off-label. In an adaptive trial design, this point's been made kind of over and over again. When you start a clinical trial, there's tons of things you don't know. You don't know the dose. You don't know the treatment effect. You don't know information about the control arm. So what is the rate going to be? We had this discussion yesterday about what's the death rate of the study going to be in advance. We don't know population. Does it work in this group of people? Does it work in that group of people? We don't know those kind of answers. Nonetheless, we have to design a trial and try to answer these. One thing that happens is as we collect information, these are things that we learn. So if we run a trial, if you run a phase 2 trial, a standard fixed trial, you may do a dose finding design. Before you go in, you don't know the dose, and then after you collect the data for a while, you're like, okay, I think this is the right dose for phase 3. In some sense, that's an adaptive process. What we do in phase 2, we collect data, we make some changes, we figure out a dose, we go on to phase 3. Those kind of decisions are adaptive. But generally speaking, when we do drug development, we only do those in between the trials. So we have the notion that we run a phase 1, we essentially close our eyes, run the phase 1, collect the data. Okay, now I know the MTD. All right, now I'm going to close my eyes, I'm going to run the phase 2. Now I know the actual dose or the actual population. I'm going to open my eyes again. Now I'm going to start phase 3. I'm going to close my eyes, and then at the end, I'm going to figure out if the drug actually came through or not. So you can imagine this, if you're driving in town, ordinarily we do not drive by starting the car, pointing it in a direction, closing our eyes, hoping we get to the next intersection, then opening our eyes and then deciding which direction to turn, close our eyes, drive to the next intersection, and so on. You keep your eyes open the whole way, and you try not to drive off the road in the meantime. Adaptive designs are aimed at doing exactly that, to keep your eyes open the whole way. There are lots of questions that you learn a lot about during a trial. If you open a trial with 4 doses, you may find out pretty quickly that the bottom 2 doses aren't doing anything at all. There is no reason to keep allocating an entire lengthy trial to all 4 doses, knowing that the bottom 2 don't work. So what you would like to do is, okay, let's get rid of those bottom 2. It's an easy decision. We'll keep allocating to, say, the 3rd and the 4th dose, and we'll try to figure out what's going on there. So again, these kind of adaptations, you may not make the largest kind of decisions, but you want to be able to react to what you know. It gets at the idea of, I didn't know a lot when I designed the trial, so I did the best I could. As I get information, at any point during the trial, you would often go, oh, if only I had known then what I know now, I would have designed a different trial. So you try to give the trial the opportunity to react to as much of that as possible. Now, do you get the fullness of every exploratory analysis? No, there's still plenty of value to looking at data between trials and really trying to understand it, but you're trying to build in efficient use of resources. So the kind of things that you might learn during a trial are that the drug doesn't work. So this is standard notion for futility. Valerie talked about this. You may find out that certain doses or treatments are promising and that other ones show nothing. You want to concentrate on those. You can find out the treatment works incredibly well. So this is unfortunately rare, but it does happen. You've seen some examples of this where you may want to stop the trial and get this treatment to patients. You could also learn that a particular group of subjects doesn't benefit. So we do lots of trials in stroke or traumatic brain injury, and it's important that patients in certain windows of time benefit from the treatment more than others, or that patients with certain severity of disease benefit more than others. You may find out that, okay, this group of patients, the treatment doesn't do anything for them, but another group of patients, the thing may be still promising. It may be valuable to try to focus your research budget. The kind of things that you can do, essentially anything that you can pre-specify in advance, you can do. I would recommend you not get overly complicated with anything, but if you can write it down in a protocol that this is what I will do in advance, it's usually fair game. So you can drop doses mid-trial. If something doesn't look good, you can get rid of it. You can also add doses mid-trial. We designed a trial for hypothermia for cardiac arrest, and one of the issues with hypothermia for cardiac arrest, at some level, it's somewhat standard of care in some places, a lot of places, but no one, it's not FDA approved. It's never actually had a good study on how long you can keep somebody cold. So we had a trial that started out, it investigated 12 hours, I may have this slightly off, but 12 hours, 24 hours, and 36 hours of cooling. And if the design found out that 36 hours of cooling was the optimal, it could open up a 48-hour window, but it had to have information to know that it looks like the dose response is going on the top edge. So you can add doses or treatments. You can stop for futility. Valerie talked about this in some detail. You can stop for success, the opposite note. You can stop enrolling certain populations. You can say it doesn't look like it's working in this group. Let's not enroll any more people who haven't gotten into the ER in 6 hours, for example. That's fair game. And you can also seamlessly shift phases of development. So there's a lot of trials these days that are either phase 1, 2. So they try to identify an MTD and immediately go into phase 2, or they're phase 2, 3, where they may try to isolate a dose. I did not do very well. And as soon as they feel like they've gotten the dose and it's promising, they seamlessly transition into phase 3. So you can do all of that. These are usually called, so futility stopping, success stopping. These get names like arm dropping or adding, so if you're adding treatments. Adaptive randomization, I'm going to talk about this in the Ebola trial, is kind of a softer form of arm dropping. If you drop an arm, you're in, and then suddenly when the next patient comes in, that randomization probability goes to 0. You don't allocate anybody there anymore. Adaptive randomization is a softer form of this, where if a treatment starts to do bad, instead of allocating it 4 arms, you might be allocating 25% of your patients to arms in a fixed fashion. Adaptive randomization says if you start to do bad, we're not going to drop it to 0, we're not going to get rid of it, but it's going to start to have 20%, 15%, 10% of the patients. And as it does worse, we're going to gradually get away from it and focus more on the good arms. The notion is just to try to not step off a cliff, so to speak, is, okay, we're going to start to work it out, work it away. If it comes back, it can come back into the trial. And then enrichment is the usual term for trying to get certain kinds of people into your study or dropping certain kinds of patient subgroups. So the basic idea here is in modern trials we have lots of questions. Dose, treatment, we have populations, we have how long should the study be? The notion is, as you answer those questions in the trial, to the extent that you can specify in advance, go ahead and do that. So here's an example. This is becoming a dated example, but it is not as dated as my O.J. Simpson trial example class when I was a college professor. So during the outbreak, there were many different treatments proposed for Ebola. So there were rinsdapivir, favipiravir, there was CMAP, there were lots of things that people thought might work, including basic stuff like replenishing fluids, etc. I'm just going to give these letters. So suppose we had 4 treatments, A, B, C, D. In reality, there were probably 5 or 6. Suppose you also are interested in combinations. So this was a big deal. This is becoming a big deal. A lot of deals and combinations are important. So not only could I give A, B, C, or D, I could give A and B together. I could give A and C together, A and D together. So I can mix and match these. Now, in reality, there was a somewhat complex structure. So you could potentially give A and C together, but you'd never give A and D together, for example. I'm eliminating that complexity here. But what I've got is, this essentially gives me 10 possible treatment arms. So I've got A alone, B alone, C alone, D alone, and any combination of the 2. So that's a lot of arms to be studying in a trial. So I want to know, well, how do I figure out which one of those 10 is right, especially given the fact that I know people are dying like crazy as I go. One option is to sequentially test one of these at a time. So I start in, I go, okay, I'm going to test A until I get an answer about A. And if A worked, I'll declare victory, and maybe I'll try to improve it. But if A doesn't work, then I'll go to B. And if B doesn't work, I'll go to C. And if C doesn't work, I'll go to D. And now I have questions about when do I start the combination. So if A works, do I then go to A plus B, or do I try B? Anyway, lots of stuff that we don't know about that. Now that's a strategy that has some advantages if you pick the right drug or treatment first. So if you're going to do A first and A is the right answer, you were lucky and you win quickly. And that's a good trial. Now the problem is, if the sweet spot is C plus D, you're going to be several months before you even get to figuring out that you've never given patients those treatments. So you're heavily dependent on making sure that you know which one is most likely. The alternative strategy at the other extreme is to give all the combinations up front. So I've got 10 different arms. Let's run a trial. You can imagine if I'm running a 250 patient trial, I've got 10 arms. I'll allocate 25 patients to each arm, and I'll let that study run. Well, these are death rates. They run about 40%. They're highly variable. If you have 25 patients per arm, there's a lot of noise in that study. You have a fairly limited sample size. The other thing that's happening is, remember something very particular about Ebola. It is very important that you treat the patients in the trial well. There's not necessarily this huge patient population where here are the patients in the study, and I'm going to have another 1 million patients out there that are going to get this later. The patients in the trial are at some level the patients who need the treatment. So we want to treat them well. If I gave 25, if I had 250 patients, I gave 25 each. If C and D is the right answer, only 25 people got the good combination. I don't know what happened to the other 225 patients in my trial. So is there any way we can bridge this gap? So we're going to do adaptive randomization. So we're going to start with subjects on all treatments, and we're not going to run 25. I think we're actually, I think in the next slide it says we start off with only 3. So a very, very small number. So we do 3 patients on every arm. Now that is not much information. We're going to look at the mortality rates every few subjects, and we're going to adjust the randomization. So when we start off, we got 10 subjects, basically 10% are assigned to every arm. We're going to allocate a few, and we're going to look at the data, and we're going to adjust those. Now remember, we burned in this burn-in period that Jason talked about. We only did 3 per arm. We are not going to change allocation probabilities from 10 to 0%. So, because 3 patients isn't enough to do that. But we can start tweaking. If we see 3 people lived on an arm, they go up, they go from 10 to 11 to 12. If all 3 people died on an arm, they can go from 10 to 9. And what we're going to do is we're going to look again in a few patients and again and again. Things that start to do well, they go 10, 11, 12, 15, 20, 50, 70% start to actually really focus. If patients are dying on arms, they will go from 10 to 9 to 8 to 5 to 2 to 0 as we go through. So we're not reacting violently, but we're trying to react continuously. So getting back to the driving analogy, we're trying not to open our eyes and have to veer the car back on the road. We're trying to gently move the steering wheel and stay in the lane, so to speak. The key things that we care about here are twofold. I want to know the mortality rate of the study. How many people live that are actually in the study? And I also want to know the chance of picking the right treatment at the end. So this is focused on the patients in the trial. We're also focused on the patients that are outside the trial. So let's see, 250 subjects. Let's make sure I got all these. We're going to burn in 3. We had a model, a generalized linear model working under the hood that actually determined the formula for going from 10 to 9 to 8 or 10 to 12 to 15. There was a formula for that. And so we ended up, we're going to change our allocation. I think it was about every week is how this was going to work. This kind of trial can run perpetually. So we designed this, an interesting feature of this different from a lot of other trials. CMAP was unavailable when we first designed this. So it had a provision that we start allocating to these 10 arms. When CMAP became available, it suddenly becomes 15 arms. And so you can gradually add drugs and remove drugs from this trial. So the trial can run perpetually. Here are results, kind of a reasonably fancy graph for results. Let me show what all this stuff means. If you look at the blue here, these are the true scenarios. So I've got my drugs A, B, C, D. I've got drugs A, B, C, D. If you see like D and D together, that just means give the single agent D. So the single agents are along the diagonal. The combination, so like D with A, is this one. The blue numbers are giving you the true death rates. So what we're trying to do is we're trying to do power analyses. If we had a particular truth, what are the odds that my trial is successful at finding that truth? So in this particular version, what we have is the true death mortality rates for anything involving D is 0.2. So 20% death rate. If you give anything else, the death rates are 0.4. So this is relatively simple. D has an effect, nothing else does anything, is what this amounts to. So what we would like the trial to do is quickly realize that D is the right answer and give those people D. The number up top is the average number of subjects that are allocated to that arm. So remember, if we did fixed randomization, we'd allocate 25 patients per arm. Here what happens is if you take the number allocated to dose D, so that's this one, this one, this one, and this one, I'm talking about, this is mortality. I'm not particularly worried if I gave somebody D and C as opposed to just giving them D. I have given them a drug that didn't add anything, but the key thing I'm trying to affect is mortality. So I'm not worried if I over-treated them, as long as I kept them alive. So you can see the average number of patients that get drug D and have this ideal response. This is 200 patients. If I had done a fixed design, I would have only given 100 patients dose D, or treatment D. So here I have 200 patients. I got twice as many patients on the right dose. The dots in here are just trying to give you a visual impression of how many patients are on each dose. So this should be 50 dots, and then 20% of them are red for deaths. So you can see I haven't given many patients the average number out here, 8, 11, so I guess 8 on the diagonal, 11 on the off. So I've gotten away from the things that aren't working and giving the things that do. The other piece of this is what do I say at the end of the trial? What treatment do I announce as the most effective? That's the bottom number here. And I've done this, so this says that 26% of the time, 0.26, I know it's small, we would say give D plus A. Now that's not the ideal answer, because we're over-treating, but it's a good answer. 25% of the time we'd say D plus B, 26% of the time D plus C, and 21% of the time give D only. So almost 100% of the time the result of the trial is to give treatment D. That's the right answer in this case to give the ideal rate. The other thing that's important about this trial is how many people died in it. So if you do a fixed trial, 100 patients get this good death rate, the other 150 get the bad 40%. On average, 80 people die during that trial. If you do the adaptive trial, more people are given treatment D, and the number of deaths is 61.6. So the trial has a good chance of giving you the right answer at the end, and we've saved 20 people's lives while we're doing it. Yeah.
Can you just explain what information does it add to you to have 4 arms with single treatment? Why does it
So you certainly could, if you wanted to only do the combinations, I think you could do that. Generally speaking, there was a lot of interest in making sure that the single arms were included. If the single arms are doing badly, the adaptive randomization is going to get away from them anyway. I don't recall a lot of discussion. I don't recall a lot of discussion about getting rid of the single arms, whether that was, you certainly could. The big discussion when we designed this trial, which actually isn't even on here, was whether or not to include placebo. Was the real controversial item. So you absolutely could. If you didn't want to get the single arms, you could start with these and then try to work your way down.
You get the same information.
Well, you've never seen the single arm, so you know D works, but you don't know if D needs something. I think if D and A worked and D and B worked and D and C worked, boy, you have to assume. That's the next thing to try.
Yeah, I think that's a good idea. I mean, because to maximize a patient getting something that works, you could probably start everyone on a combo. Yeah, and if D plus everything were kind of the same, we're like, oh, once you get D, nothing else matters, and then almost go back to D. So in terms of making sure patients get some medicine that might work, it might be a good idea. Of course, there's dosing issues then because you're now doing combo drugs and such.
Often in clinical trials, regulatory agencies require a single arm so they can attribute the side effects to the single arm. In combined arms, you can't be sure which one is responsible for the side effects. But if you have well-established agents, you might not need to do that as here.
I think the summary is it's a reasonable thing to consider. Let's see, so let me skip this one. We tried to do this under a variety of scenarios. So here's another one, this is scenario 3. This is a situation, dose or treatment C has some small effect. So death rate's 40%, C takes it down to 30, D takes it down largely to 20, but there's a little bit of synergy here. So if you can give D plus C together, that takes it all the way down to 10. So if you do this, you can see again, patients are allocated mostly to D. So we've got 34, 33, 89, 25. So that's again, 200-ish. And also now you've got a decent chunk, you've got 90 patients on this optimal arm. The other thing that's happening, so what treatment did we pick at the end? The adaptive trial said give D plus C 89% of the time. So that's at some level the selection probability. It's not even the power, it's a little bit stronger than that. Did you get the right answer? If you had done the fixed trial at 25 per arm, then that probability is only 82%. So we have a higher chance of giving the right answer. Now, the other way that you could do a fixed trial is, again, the strategy of, oh, I'll go in with one, and if it doesn't work, I'll go to the next one. The difficulty with that is if you pick D plus C first, that's great. You'll actually do better than this. But if you pick D plus C last, it takes you a long time to get there. Whereas this one, again, figures it out and saves about 20 patients as we go. This is scenario 4, just a little bit stronger version of this, where D plus C is still at a 10% death rate, but the other ones are worse. So it makes it more of a sweet spot. And again, you find it, you really pile patients there, and here you're saving 40 lives. All right, so the morals of this, the trial treats the patients in the study better. So I know Will has a chance. Will here. Okay. All right. If you weren't here, I wasn't going to say anything nice about you.
Are you going to talk about my Ebola suit?
No, I was going to actually. I'll allow you to do that later. I was going to talk about your JAMA paper.
Okay.
So anyway, Will has a nice article on adaptive randomization and the advantages in recruitment and patient preference as well. So I'll plug for that. And also, so you're not losing anything in terms of this adaptive randomization. It's not costing you performance. We're more likely to pick the right treatment. And the reason that we're more likely to pick the right treatment, in the examples I showed, A, B, and C didn't work. I get rid of them, and I pile all my patients on the interesting arms, D plus A, D plus B, D plus C, and D. So I figure that out, and I focus my resources on the question that's left at any given time. All right, so a bit of a switching gears here. I gave you numbers. I said, here's the probability that you pick the right dose. How did I get those numbers? There is not a formula, so you can't go on to your favorite statistical sample size estimator and go, tell me for an adaptive design with 10 arms, what is the probability I pick the right dose if I do it this way? That doesn't exist. There are simulators. So what a simulator does is it basically runs the trial virtually over and over again. And so that's how we address this question. So to get an idea about what a simulator does, I'm going to do the absolute prototypical nerdy stat professor example, which is we're going to flip coins. So if I flip a coin 10 times, anybody know what the probability I hit 4 heads in a row is? You want to take a guess? Will, what do you think?
30%.
You were here last year. So it's about 24.5%. So I did not know that offhand. You can actually, there are mathematical formulas, you can derive that. Put in some matrices, you can get the right answer. So with some complicated, not so complicated math, you can get the number 24.5%. The other thing that you can do is, if you have a coin, you can sit and flip it 10 times and determine whether or not that happened. And you can repeat the experiment over and over again. Flip the coin 10 times, did it happen or not? Do it again and again and again. So here are particular examples. My simulation 1, I got this sequence, no sequence of 4 heads, so that's a no. This next one, I got a sequence of 4 heads, that's a yes, and on down. If I do this repeatedly, and thankfully computers are willing to do this as often as you want, I can get a darn good estimate of what is that rate without having to go through the complicated math. It's a pretty trivial, do this thousands of times and just count the proportion that one. Preview of coming attractions, I'm going to do the exact same thing with the clinical trial. I'm going to simulate the Ebola trial. I'm going to basically do coin flips for every patient, whether they lived or died. I'm going to fit the models. Everything that I would do in the trial, I'm basically designing a flight simulator. If I run the trial this way, here is the chance that I'm going to pick the right dose. And I'm just going to add those up. The number of simulations you need typically is well within what a computer can do. It depends on the number you do. I did 100,000 and got a rate right about 24.5%. If you can do 100,000 simulations, you can estimate most things within a percentage point or a lot less than that, actually. So you can get quite good accuracy. Another kind of question that I could ask is, how many times do I need to flip the coin in order to give me a 90% chance of such a streak? So if I flip it 10 times, I've got a 24.5% chance of getting the streak. So if I flip it 50, does that give me a 90% chance of the streak? If I flip it 100, does that give me a 90% chance of such a streak? So I don't know the answer to that one either. So, and that one, there's not really a formula for that I know of. That will just tell me this. I can still use all the simulation methods. So I just kind of can randomly search. I know what n is equal to 10. I got a 24.5% chance of it. I tried n equals 50, did the simulation, 82.9%. I went up longer, n equals 70, got 91.7. Oh, that's too high, and I worked my way back and forth. This is the kind of calculation you would do to find power. If you want 90, what sample size do I need for 90% power? Well, we're going to simulate a trial with 10 subjects. That's only 24.5% power. We'll simulate a trial with 70. It is a fair amount of trial and error, but you can binary search this and you can get to the right answer relatively quickly. All right, so back to reality. Generally speaking, complex adaptive trials, there are no formulas. So there is no complicated math that allows you to check your answer. The only thing that you have available to you is doing these large-scale simulations of the trial. And that means simulating all of the individual patients, all of their outcomes, when they arrive. So you simulate for accrual rate, and when do they arrive. For each of the interim analyses that you want to do, here is the random data that will arrive at a particular time. Here's the model fit that I would use. Here's the adaptive randomization. This can get very, very involved on how this works. I'm going to do a fairly simple one of these to kind of give you an illustration. This is a trial with just 2 doses and a placebo. So 2 active arms, placebo, low dose, and a high dose. I'm planning on running a 216 patient trial, and I'm going to enroll 36 patients per month. My endpoint is a composite. So the key thing here is my low dose may very well be the best dose. Composite endpoint, you can imagine the high dose gives me great efficacy. You could imagine the low dose avoids the safety part of the composite. So the low dose may actually be the optimal dose. And I also am in a situation where I'm really considering, on whatever endpoint I'm using, increases of 2 units are considered valuable here. If I do a fixed trial on this, so I'm going to enroll 72 subjects per dose, so control low and high. Remember what Michelle told you about multiplicities. You've got 2 doses, you've got 2 shots at the apple, so we need to do a multiplicity adjustment. I'm going to do a Bonferroni, I'm going to cut my alpha in half. If the drug doesn't work, so this is a situation where these are changes. When I say true effective low, this is the change from baseline. So the true effective low is nothing. It's the same as, sorry, not baseline, placebo. So the true effective low is 0, same as placebo. The true effective high is 0, same as placebo. There's nothing going on. In that case, what happens, a fixed trial gives you a type 1 error rate of about 2.3%. That's the probability of trial success. And in that case, it's an incorrect success. So what's going to happen is if your drug doesn't work, you're always going to enroll 216 patients, and the most likely answer is you're going to fail. So that's unfortunate. We're going to talk about futility is what we're going to simulate. If the drug does work, then what's going to happen is the true effect in low, this is hypothetical, suppose that is 1 and the true effect in high is 3. That's a good drug, and it's important that I pick the high dose, because that one is much better than the low dose. In that case, the probability of success is 77.1. Plug this in the standard calculator to get that. All right, so here is a naive adaptive trial. So I'm not making this complicated, so it's naive from the standpoint of I just wrote something down. After 1 month, so here's my futility analysis. After 1 month, I'm going to stop the trial if neither dose achieves a 2-unit increase. So that's what I want. So I'm just going to take a quick look at the data. If I don't have it, I quit. After 3 months, what I'm going to do is I'm going to choose the dose with the higher observed mean. So after 1 dose, I've got this quick out. After 3 months, I'm going to pick a dose and I'm going to get rid of the other one. And I'm going to keep enrolling the trial. So I'm trying to focus, this is a dose selection, focus on that. So examples, the first trial, you can see the month 1 futility. This is my placebo mean, 6.4. The low dose is 4 points higher. That's good. The high dose is 3 points higher. That's also good. I'm going to continue my trial. I get to 3 months. The data's gotten a little bit worse. That randomly happens. And what happens is the low dose looks a little bit better. So I'm going to choose the low dose and get rid of the high one. And now what goes on is at month 6, I'm going to do my final analysis. Things have continued to get worse. Again, may just be noise. This dose is out. I do a statistical test on these. I haven't shown you the variation. I try to keep the slides simple. And I've got a success, and I've got a p-value less than 0.001. So that's a nice successful trial. Example 2. Here's a trial at my month 1 futility. I had no effect on low. I had a 1-point effect on high. My rule says to quit. So I've done something important here. I have told you in advance how I'm going to run my trial and what the conditions are. At month 1, here is exactly how I'm going to quit. At month 3, here's exactly how I'm going to pick a dose. And at that point, you can let the computer autopilot the trial.
Questions?
Yeah.
So are you using your alpha in the first code computer just a qualitative look at points higher and how you're going to proceed? Or are you going to test and compare the parameters?
At month 1? Yeah. At month 1, no. I mean, it's naive. I just wanted a 2.9. I do not recommend that, but it's easy to describe what it is. But there's no test. The adjustment that I'm going to make, I am going to put statistics on it. If anything, I did this so I can illustrate how bad it was. All right, so let's see. I'm going to simulate 1,000 trials under the assumption that the drug doesn't work. If the drug doesn't work, what do I want to have happen? I'd like to get out at month 1. If the drug doesn't work, it's likely to fail. I might as well use my resources for something else. This is a simulated plot. This is the range of data that you would see over 1,000 simulated trials. So the true effect, the low dose truly has no effect. The high dose truly has no effect. This is the right, true answer. Sampling variability is very big. I'm only a month into the study. I've only enrolled 36 subjects. That is 12 on each dose. You can see there's a wide range of variation. So for example, the low dose, you could get answers that are -5, -6, -7. You could also early, it looks like the low dose is on the positive side, 5 or 6 or 7. So some of those trials are going to give you dramatic, this is killing people, dramatic, this is working incredibly well, and it's all noise. So if anything, the right conclusion here is, wow, 1 month may be a little too early to make this decision. The decision rule, and the high dose does the same thing. The decision rule that I had said I needed at least 1 dose to have a 2-point effect. That was my rule for continuing. So the black dots here, these all have a 2-point effect. These black dots, the low dose gets me at least the 2-point effect. These black dots, the high dose gets me the 2-point effect. They're all continue the trial. The red dots, these are the trials where neither dose got me my 2-point effect, and I declare futility. So I can just count, just like I can do the streaks in the coin flip, I can just count how often do I declare futility. And it's 679 of the dots are red out of 1,000. So that's great. 68% of the time when the drug doesn't work, I'm going to only use 36 subjects in my trial. Not a bad futility rule. Get out often and early. There's a flip side of that. I want to get out of the trial if the drug's in fact bad, but I don't want to get out of the trial if the drug is in fact good. So if I have a situation where my drug actually works, remember how much random noise we had in that system. If I have in truth a good drug, it can have a bad first month. And I don't want to go, you had a bad first month, I will give up on you forever. So this is what the data looks like when you have a good drug. The true effect here for the low dose is 1, so the truth is here. The truth for the high dose is 3, that's about here. So in truth, everything works. This is the same range of variation, but of course it has a higher mean because the drug in fact works. The points are still colored. These are the trials that you stop. So number of red trials, that's 305 out of 1,000. So that says that even if your drug is effective, with this futility rule, 30% of the time you're gonna quit. That's an incredibly bad average. You're setting yourself with dramatic loss of power. This actually happens. This isn't just a naive rule. I was reviewing a Pcori grant, which I won't go into details about, but in any case, they had a futility rule where they said every month they were going to look, and if a particular condition held, they were going to stop the trial. This was their safety monitoring. It's good to do safety monitoring. However, I sat down and I did the calculations and I said, suppose there's no safety effect whatsoever. Well, what's really going to happen? In theory, it's all flat. In practice, it's going to go up and down at random. If you're looking every week, every month, I calculated what are the odds that at some month during the trial, I'm going to make a bad decision. And the chance that they would stop the trial, even with no safety signal whatsoever, was 55%. So, and the rule made perfect sense if they were looking at it once. It doesn't make sense if they're looking at it multiple times, knowing that multiplicities are going to happen. So this is why it's important to understand your futility rules. As Valerie was talking about, also related to the multiplicity issue from Michelle was talking about. So in this trial, this futility rule is way too aggressive. And to kind of see what the issue is, I've color-coded the points here. The green dots, these are the dots that I got when the drug worked. The purple dots, these are the dots that I got when the drug didn't. So they're the same dots I showed in the last few slides, but now I show here's what the bad drugs do, here's what the good drugs do. And the key issue, the good drugs are green, the bad drugs are purple. These overlap like crazy. So you can get a bad drug that looks like a good one at the early interim. You can get a good drug that looks like a bad one at the early interim. You cannot tell these apart with any accuracy. If you wanted to put on a futility rule at 1 month, you would need to put it down about here. This is not necessarily a bad rule if you did this. It would be very conservative, but at least you know you're only eliminating bad drugs. You're not eliminating the good ones. So you can calibrate. So I changed this a little bit. I set up, instead of just having a conservative one, and again, you could set up all kinds of monitoring systems. This is just a for example. I decided to look later. So instead of looking at 1 month, I was already doing a dose selection in 3 months. Let's move the futility forward. We're only going to do futility in 3 months. Now, when I stop, I don't save as much. I only save half my trial rather than most of it, but I'm hoping to be a lot more accurate about doing so. I'm also going to change, this is what you were getting at. I'm going to change my rule to something more statistical. I'm not saying this is a great rule either, but it's at least statistical. Again, I was trying to pick something simple here. I'm going to pick a rule that says I'm going to be futile if neither dose has a p-value less than 0.25 compared to placebo. This is a situation where I'm halfway through the trial. For me to win at the end, that p-value has to get to 0.025. Halfway through the trial, I need to be on my way to 0.025. So if I don't have some kind of decent signal, I'm going to be out. Whether this is the optimal p-value, whether p-values are the right thing to do, I'm not making a statement about that. I'll take a longer discussion. But that's what I picked in order to show how you would think through the problem. So at 3 months, this is what the situation looks like. If I consider the range of variation, these are good drugs, and I've simulated 1,000 trials. So in truth, they're at 1 and 3, and then of course there's still variation. So some of them are high, some of them are low. The bad drugs are still in purple. The dots here, the dashed lines, this is my futility rule. So I had a rule that said your p-value had to be less than 0.25. I back solved that. What treatment effect do you need in order to get a p-value less than 0.25? And it turns out this region right here, all of these trials are considered futile. This is a much better rule. The purple dots are the drugs that don't work. I actually get 62.6% of those. So of the ineffective trials, 62.6% of the time, I end the trial in 3 months. The nice thing is, I hardly ever catch a green dot. So it's hardly ever is a little bit strong, but I only get 5.6% of the green dots. If the drug is working, I'm likely going to get past the futility. If the drug is not working, it's likely to catch it, which is what you want in a futility rule.
Well, and Kurt, is it true that the green dots that fall below the line that you're killing anyway may not be successful trials? They're successful drugs, but... Okay.
So that, go ahead and...
Well, maybe you were going to say that night.
No, go ahead.
You're right. So, if you design a trial with 80% power, you're accepting the fact that 20% of the time, even an effective drug is going to not be statistically significant. And so, in this case, you're not even, it's bad to say, oh, this is a bad designer, there's a little bit of a bad thing in it because I'm killing a good drug, when in fact that's a trial that would have been negative. And so by stopping a trial for futility that would have been negative anyway, even though the drug's good, you're not really doing much harm. You're making the same decision sooner than you would have later.
All right. The other aspect of this that I can do by simulation, there's a dose selection. I know that the high dose is better than the low dose. How often do I get the right dose? So these are the 1,000 simulated trials when I have a good drug. Not particularly worried about what dose is selected when I have a bad drug, because those are likely to be failed trials. Negative trials, anyway. So this is the same, these are the pattern of variation that I see when the truth is 1 for the low dose and 3 for the high one. So again, got a range of variation. It can go up to 5, it can go down to -3. The high dose goes from 0 to 6. So a lot of variation here. I said that I was going to pick the dose that had the higher observed mean. Whatever dose looks better at 3 months, I'm going to go with that one. So I can just count how often do I pick the high dose. This is the line of equality for my 2 doses. All of the dots above this line correspond to a trial where the high dose was doing better. and is thus the one that's selected. All the ones below the line, that's the situation where I pick the low dose. So I pick the high dose 871 times. I pick the low dose 73 times, so that's good. I have relatively high accuracy to get the correct dose. And I also know that 56 of those points were futile, so I don't pick any dose. So the final characteristics here, this is, I haven't had to do any math except to randomly generate subjects and run the rules. And I did this 1,000 times. So now I can just tally which dots ran into different things. So I simulate the data up to 3 months. I simulate the rule that I would choose. I simulate the data up to 6 months. And I decide did I win the trial. And I just tally that over and over again. What happened in trial 1, what happened in trial 2, trial 3, trial 4, up to trial 1,000. For ineffective drugs, what happens is I had 626 failures, negative trials, at 3 months. That was my futility. I also had a bunch of trials that picked the low dose. 11 of those ended up successful. Remember, the drug doesn't work here, so these are your type 1 errors. 11 trials, low dose successes. 9 trials were high dose successes. And then I have a number of trials which were negative at the end. So I can compute the type 1 error rate from this trial. How often did I have a successful trial when the drug didn't work? That's 20 out of 1,000. That's 2%. Ordinarily, the estimated type 1 error rate, you'd run a lot more than 1,000 trials, but I didn't want to plot it for the dots. If you had an effective drug, so this is the true effect of 1 and 3, remember I stopped 56 of the trials for futility. So that's a fairly low rate. As Jason said, a lot of those might have failed later if I had let them go anyway, so that's probably a good thing that I stopped them. I had 70+ trials that picked the low dose. So this was 25 of those ended up being successful, 48 ended up being negative. Most of the time I picked the high dose. And 795 times out of 1,000 I got a success on the high dose. That's the right answer. And then 76 trials I picked the high dose, but it didn't pan out at the end. Or after. The power of that trial is the proportion of times you were successful. That's those two numbers together, 795 plus 25. So the power of the trial is 82%. Now, one thing that's worth noting here is the 25 low-dose successes, that's not the same kind of success. A high-dose success, you're going to give the right drug going forward. A low-dose success, in truth, that drug only has an effect of 1. Sometime in the future, your post-marketing study or whatever, you're going to realize that drug isn't as good as you thought it was. So at some level, that is, you got the answer that the drug worked, but you picked the wrong dose. We often call that a type 3 error. So the 25 of those, one nice thing about simulation, I can tell you how often that's going to happen. If you go through a standard power calculation, a lot of times they don't break it down to this level of detail. And here are all the different possible things that can happen and how often they occur. So let's see, my final slide, I think. Good. A comparison to fixed trials here. The type 1 error rate was 2.3%. We think it's now about 2%. It should be a little bit lower. I can say I haven't estimated that perfectly, but it's a little bit lower. The power has gone up. So I started with 77.1% with the fixed trial, and now I'm at 82%. I've gained power by doing this. I gained power by focusing the second half of the trial on the dose that looked the most promising. So I got more patients on it. Now remember, that 82%, I've got some of those are type 3 errors, but also remember some of these are type 3 errors as well, and I haven't told you how many there are. But so this number, the 5% increase in power, is not diluted by that. Type 3 errors and type 4. If the drug doesn't work, do I get futility savings? I ran a fixed trial, the answer is no. I always ran it to 216, never saved anything. If I put on the futility rule, I saved half the study 63% of the time. So not always, but I got a decent chance of getting out. And finally, there's an issue of the sample size on the selected dose. In the fixed trial, I have 72 per dose. When I did my current study, I eliminated a dose halfway, and I only allocated 1-to-1 rather than 1-to-1-to-1 for the second half of the study. That gives me more patients on the dose that I actually end up getting to the end with. So that's important for safety analyses and just better precision of estimates when you get to the end of the study. All right, I am out of time. So conclusions. Adaptive trials allow you to change information or change the trials based on incoming information. And keep in mind, you'll need to simulate these to figure out how it works, but a lot of times the simulation exercise can really let you see fine detail that can really control what happens. And there are plenty of people and software available to help. Thank you.
Does increasing your trial efficiency come at the cost of reducing the risk of reaching that statistical significance because of multiple testing, or by monitoring how well
So I think what you're asking is, are you asking is because I've done various looks and changes, have I somehow introduced extra multiplicities or something that cost me power? So it depends on what kind of adaptation you do. In this particular case, actually both of the adaptations that I did actually helped. I did a futility analysis. So, and I said, well, the answer, I think the right answer is complicated. The futility analysis actually cost me some power. By not allowing all the trials to go to the end, it's a little like if you're at a basketball game and suddenly your team's down by 20 points at halftime and you leave. A lot of the times you just got out of the parking lot earlier and you didn't have to watch your team get killed, but there's always some come from behind winners where you missed the, oh, we made a 25-point comeback. I don't allow that to happen. So that's a little bit of a cost of power. I can quantify how big that is. So this is what Jason was getting at, is I could count those green dots, how many of those actually were come from behind. It's really small. The other aspect is the fact that I selected a dose and went forward. That has some interesting, in terms of the multiplicity involved. If I had let both the doses run to the maximum sample size, I've got to worry about the multiplicity that I've got 2 doses in the study. If I go halfway through the study and I stop one and only have one go to the end, I still have a multiplicity, but it's less of one because I focused in and I let the trial run past the time of my selection. So I think my answer is that you certainly there are pros and cons of doing each of these, and the purpose of the simulation is actually quantified. So there's nothing more secret. When I got the 82%, all of the pros and cons of every one of those adaptations is included in that 82%. There's no secret adjustment that you have to do at the end. If I simulate it the right way, it accounts for all of the pros and cons, and you can investigate it in more detail how they occur. Yes?
If I just correctly, I mean, the assumption was based on a minimum effect size or a minimum clinical effect. Is there a way of factoring in actually more than that? Like, I mean, if you have an effect of 2, 3, 4, 5, so basically as opposed to a binary having a like or even sort of continuous way of assessing the effect size, so you can measure more than minimal effect.
Yeah, absolutely. So you could base your rule on the full distribution. You can base, there are the kind of rules, you can kind of base them on anything. So if you wanted to have a more, a less strict kind of, line in the sand, 2 or -2, you could certainly incorporate that into the trial. You could take into account the effect of the drug in terms of when you can change your sample size. If I had a huge effect at 3 months, I can say I'm only going to go, I can stop now, I can only go to 4 months. I may decide, look, I got a huge effect, I don't need to collect all the data out to 6 months. I can collect out to 4 months. All of it.
And perhaps, just deciding between the two doses, so if I have got a sort of significantly different effect size in one dose compared to the other, does that allow me to sort of do the selection more effectively and sort of move medications to one dose?
Yeah, so you could look for that dose selection at more than one point. Suppose if at 3 months, both of the doses are doing about equally well, you may decide you don't want to get rid of one of them. You want to keep them both in and try to learn about both. So you could do that. You could also set up a rule that, maybe 1 month into the study, if one of the doses is doing great, another one's doing terrible, I may do the dose selection earlier than at 3 months. So you can certainly take into account if you get a big effect early, you can make the adaptation early. And if the effect looks, I don't know what it is, then you can certainly delay it until it's the right time to do it. Bill?
Yeah, Kurt, I think one thing would be interesting, I'm interested to hear you discuss. Although you talk about using simulations and adaptive trials, we think there's probably a lot of value in using that for fixed trials as well, because there's a lot of studies, for example, where, and we see this happen all the time, where let's say you assume your control group is going to have a 50% bad outcome, that you're looking to improve by 10% to a 40% bad outcome. But when you actually do the trial, they really have a 40% bad outcome in the control group, so you're really looking for a 30, and things like that can go on. And sometimes, if it's a long study, sometimes the outcomes in the placebo group might improve over time just because of improvements of care. You won't know how that's going to affect your outcome unless you simulate that. I'm just interested in your comments on that.
My comment is to agree with you completely. So we simulate trials for fixed trials, things that you can do the calculations, and it's very relevant in terms of not only if your control group drifts, missing data patterns. So how are you going to handle missing data? What if it happens in a particular way? It's very good to stress test the design. Simulation, you can do a lot of this analytically. But a lot of these calculations, it's easier just to go, okay, let's change the dropout rate, let's re-simulate, and really get an idea of how robust the design is to those kind of changes. And what assumptions, it really gets at what assumptions do we really need to care about, and what assumptions, okay, it's going to work no matter what. But if this happens, you're in big trouble.
Comment about using prior knowledge or prior trial data towards this kind of approach, towards selecting an agent, for example. Talk about the pros and cons of that.
Yes, I can talk for much longer than I. So at some level, it's a question that you can do adaptive designs and incorporate this historical prior information. You can also do fixed trials and incorporate the information as well. A lot of times a study you either have prior data from a similar drug, or you've got prior data on the control arm in a study, or anything else. A lot of times you can get a jump start on your current study by incorporating that information. You essentially hedge your bets into the assumption that your historical data is on point. To the extent that historical data really is on point, what tends to happen is you decrease the variance of all your estimates, and you could do adaptations earlier and more accurately if you incorporate. It really gets at the notion of take into account the totality of the information, which includes everything you do in addition to the accumulating data. The risk of it is if for some reason that historical data wasn't on point, so I do tons of antibiotic resistant stuff, which needs to pay attention to everybody here. But in any case, one thing that's big about historical data, success rates over time have tended to drop because things are becoming more resistant over time. So if you use that historical data, you can incorporate biases because it doesn't, you think it's going to be here, and in reality it's down here. And so that bias can happen when you try to incorporate the historical data. So I think the general answer is you have to substantively, is it applicable? To the extent it is, everything can be done earlier and better. To the extent that it isn't, you can bias yourself and make incorrect decisions.
Question. Is there any specific minimum number to this adaptive trial can be done, or can it be done with a small patient population?
So we have done, I mean, it depends, the same kind of questions that would happen for a fixed trial. I mean, if you can do a trial with 10 patients fixed, oftentimes you can do it a little bit smaller adaptive. So I don't think there's a minimum sample size for an adaptive trial, but there's a minimum sample size to run any trial. So we've done oncology studies, for example, in rare tumor types, and those often run, maybe 10 patients per subgroup. And we certainly do those adaptively. Now, at the same time, we're looking for big effects in those that those sample sizes can discern. So I don't think there's a sample size where, oh, this is too small for adaptive. There's definitely a sample size where it's too small to do anything.
So I just was wondering if you could make a quick comment on your simulation scenarios, how you came up with those, and how the clinicians were involved in coming up with those.
Yeah, so Michelle's getting at the point that I did cheat to a little bit. I only simulated if the drug didn't work, and I simulated if the drug worked under a particular condition. In reality, what I should have done, I do it my real now, I promise, is I should have also simulated under scenarios where both doses work identically well, where the low dose is better, where one dose causes harm. I should have considered a wide range and really tried to get an idea of the robustness. A lot of times you can see certain designs work really well for some scenarios and not as well for others. And you really want something that works well no matter what you're going to be faced with in practice.
And I guess also the point that it's not the statistician alone deciding these scenarios. You work with the clinicians and really have a ton of input on what they think may happen and what the scenarios would be. So this isn't something the statistician just goes off in isolation.
Yeah, if you're ever going to design an adaptive trial, this isn't a situation where you go to your statistician, drop off 5 numbers, and 2 days later come back and pick up a piece of paper. This is okay, what aspects of this trial do you really want us to track? Because we're going to give you fine-grained detail. What scenarios do you think are important? This becomes a real collaboration. I've always found it amusing when we do this. Often the clinicians really get into it eventually. And we start off with a relatively simple adaptive trial and we end up with, oh, well, can we do this every week? And can we add this? And can we track these other 17 variables? And so like, whoa. But we certainly welcome suggestions like that. So be involved in your adaptive design. I'm taking up a break. Awesome.
All right, thank you.